Recording has begun now. Sorry about that.
So I'm crossing my fingers that I can word this reasonably well.
Sounds
It
exciting.
talks about in the paper a bunch of these fungi that are being sequenced by molecular techniques, and to the best of my knowledge, that's also a lot of what microbiota research ends up relying on is just the DNA samples
Yes.
that we're getting from large swabs of the entire thing. Is there any proper methods to really distinguishing the individual bacteria and fungi that you're finding in those samples? Do we ever end up having one of these unknowns getting a positive identification later by like culturing methods or I don't know, observation?
Yes, in very rare cases. Because think of what that would entail. Um, Time and effort wise. You've got your sequence data, you generate your report, test your hypothesis, uh, publish your results. Then, do you have the time, funding, and energy and student power to go back to those same samples and try to then culture stuff? Maybe. You might. And if you do that, um, and get lucky enough to be able to grow something and then resequence it and then compare your original data to it, you might be able to get that confirmation there. And so it does happen in rare cases. Um, I accidentally had it happen in one of my own studies, but that was like a very reduced microbiome on some plants. Nowhere near as complex as like a human microbiome. And so I was kind of set up for success in there. And I got lucky in that the thing I was looking for was actually easy to grow in a petri dish. It didn't necessarily have to be the case. So that is something that can happen. I don't say that it happens very often. It's quite rare. Another thing that you can do is, in order to publish a paper using kind of this DNA technology, almost every journal requires that you make your data public. So all those DNA sequences, those millions of reads, they have to go online onto a repository. And so they're all sitting there. And so you can go back, and they're publicly available, and reanalyze people's data with like a new database. So you can use advanced databases that have more species in them and reanalyze somebody's data and maybe get better identifications in the future than they could at the time. But
So
that takes a lot of time and effort too.
So I assume that identifying them as such would be useful to an extent.
Maybe.
Maybe. Hmm.
Maybe. It would take some convincing to get funding to do that. You'd have to have a good reason for thinking that these particular species would be useful to identify. So if you noticed some unknown fungus DNA was very strongly correlated with a disease, then yes, it would definitely be useful to go back and try to culture that, figure out what it is, that kind of thing. But if you just found 3,000 new fungal species, nobody cares. As sad as that is, microbial ecologists are so used to finding new species that it's annoying to us. And for better or worse, if I find 400 new fungal species in a study with students here at UVU, we just delete those data. Essentially, from our analyses, we say, oh, we don't know what these are. We report that we found them. Here's the sequences if anyone cares, but we can't do anything with it, and we don't have 15 years to go and try to identify all these.
You don't at least name them after each other?
You could, but that would take a lot of work. Describing a species is not a trivial thing. You have to get it into culture, do electron microscopy on it, describe, whether it's got a Spitzenkörper or not, what the shape is. You got to get DNA sequences of several different regions, get good pictures, build a phylogeny, find out where it goes, prove to people that it does belong there, and then you get to name it. That's like probably a 2 or 3 year process.
Yeah. Okay.
I did it once in grad school, and I named a new slime mold after my friend for his birthday. And it was so much work that I will never do it again.
Hey,
it's
Dr.
important work, but it's like thankless hard work. Yeah.
Hey, Dr. Don.
Yes.
So in the paper, it talked about how the regions or the genetic loci containing the 18S, 5.6S, and then the 28S were chosen, but I couldn't figure out why those specific, like the 18S and the 5.6 and the 28S, why were those important?
It's 5.8S.
Okay.
But yeah. Why are those important? Everything has a small subunit, right?
Yeah.
Every species on Earth. Without ribosomes, you can't make proteins. Okay, so ribosomes are very important. If they mutate too much, they cease to work and you die. So it's a highly conserved gene. So what that means is that we can design primers for it and be relatively certain that the primers are going to work in a bunch of different species, because there are regions of that ribosomal encoding DNA that really just can't change, and they're essentially the same in bacteria and humans, because we all have ribosomes. There are some differences, and so in fungi, we've got primers that do a pretty good job of getting essentially all fungi. They miss a few different groups, and so this is known, and we try to account for that bias by using like different sets of primers and combining the results, or just mentioning that in our analysis, like, oh, the primers we use probably aren't picking up any of this group, so take it with a grain of salt. But inside that small subunit, there are regions that can vary, and they do quite a bit. So let me share my screen so everyone can kind of see a picture of this.
So, Dr. Zahn, so that's why those ribosomal heads or subunits are the main targeted genetic regions, just because of like, are you saying like consistency?
Yeah.
Because like no matter mutations, you can't mutate those subunits, because then they just cease to work, which means the cell will die.
Yes.
So those are always consistent then.
Yeah, and highly
Okay,
conserved.
that makes sense.
Yeah.
Gotcha.
So they're highly conserved. Resources. So I've got up, this is the Unite database. This is the most commonly used one for fungi. Let's see if I can find the primers. Unite primers. Ah. Okay. So this image here is showing pretty well vetted and commonly used fungal primers, but really what it's showing is the small and large ribosomal encoding DNA subunits. Okay, so the 18S region is pretty important and has a very conserved function, and so we can design primers that sit in there, and we know that for most species that region is going to be the same DNA code. Okay. But right after it is this ITS1, this internal transcribed spacer, and it is totally unimportant the sequences there. Its only function is to have a space. It's not like protein encoding region or anything. It's just there as a spacer. And so it's allowed to mutate freely. Okay. And so we like to design primers that begin here at the end of the 18S and amplify this ITS1 region, or design primers that begin here in 5.8 and amplify the ITS2 region. So you can see these are named forward and reverse primers. So given that current cheap DNA sequencing technology lets us get, at most like 300 base pair long reads, that's what we have tended to do. It's just amplify either the ITS1 or the ITS2. Now, these are pretty different between species, but the same within species. And so we've just found that the ITS regions for fungi make a pretty good code for the species level. It's not perfect, but it does a pretty good job. So if something has the same ITS1 region, it's taken as a safe sign that it's the same species as something else. Did that clear it up for you or make it worse?
No, that makes a lot more sense. So I was just curious why those specific ones were chosen. It didn't really talk much about it, so.
Yeah, it really didn't. I'm glad it talked about it some, and I was hoping this question would come up, but yeah, it's just a matter of, we know that we can have more or less universal forward and reverse primers for PCR without knowing anything about the ITS1 and still get it amplified.
Perfect, thank you.
We do the same thing for bacteria also, but they have 16S instead of 18S.
We covered it a little bit before, but is there a reason why when we give antibiotics and, it destroys a lot of our microbiome, which allows the fungi to get a better hold, why don't we give some antifungals along with that to kind of preemptively prevent that?
Because we don't know if the fungi getting a hold is good or bad yet.
Simple enough.
Yeah, I mean it's the ethics of do no harm for medicine, right? You don't want to go experimenting willy-nilly with that.
Benjamin, you had your hand up before me. Did you want to ask a question before I ask mine?
Oh, I missed that. Sorry.
Yeah, there we go. Sorry, my mic takes forever to click back on. No, my question was about Candida. It just seems like it's all over the place, all over your body, within your body, in your GI tract. Is that all one fungi, or is that like different branches of it?
Probably the same.
All the same. So a follow-up question: if it's all over, and it says that even healthy people have a ranging up to 50 to 65% among hospital workers. So why isn't that more of a concern? As far as like, is it just not as dangerous? Because I did a little bit of back research on it, and it just seems like it can get pretty bad. I mean, it can even cause death, can it?
Yeah, but in most scenarios, it's actually really good.
Okay.
It's a fine balancing line. It can be pathogenic, but more often than not, it's actually mutualistic. Beneficial because it's not doing any harm, but it's taking up space.
Okay.
Taking up that niche to where bad things would be showing up. And so Candida, like Candida albicans, which is all over our skin, right? Yes, it may cause rashes if it's kind of getting out of control on you. But more often than not, it's actually just living there, eating your filthy skin, and taking up room where bad bacteria would be landing and growing. And so in that sense, it's actually beneficial for us because it's preventing other things from growing that we don't want, and it's not doing any harm in the process. Or if it is doing harm, it's something like a light rash, or maybe just some dry skin. Yeah.
Okay, that makes
What
sense.
about
Thanks.
yeah? Could there be any like resources that the bacteria is getting from the fungus?
Maybe. I hate that I have to keep saying maybe all the time, but this is mycology, and you just get used to that answer as a mycologist. We don't know. More research definitely needs to be done on that. We don't have a good solid answer right now. As Hayden was asking, like, yeah, this is still kind of new, and the best review is from 2013, and we haven't actually advanced a whole lot in that time. I mean, microbiome research is pretty new. It's only been enabled by, high throughput, deep DNA sequencing. And so as those tools become cheaper, we're starting to get more answers and starting to get actual hypotheses to test out. So it's kind of exciting if you're a scientist because there's all these unanswered questions, right? It's not like a Newtonian physicist. You already got all your answers. Sorry. Like, yeah, you know the relationship between mass, acceleration, and force. Good luck overturning that. But in mycology, especially with microbiome research, it's wide open. There's a lot of room to contribute new information. All right, see a hand.
Jonathan had a question in the chat. He said, the paper says that there is no strong evidence of a mutualistic relationship with the fungal microbiome. How has this, or has this significantly changed since 2013?
Yes, in the candida on our skin sense. So we know that it can be pathogenic on us, and candidiasis is also known as thrush. Right, so it starts growing in your mouth. That's not great. It can start eating you, but mutualism is not always mutualistic. It's always teetering back and forth between being a mutualist or being pathogenic. It's not necessarily a stable relationship that we have, but we do have strong evidence now that candida is, or it tends to be mutualistic in most people. As far as other species, no.
And is that just, when you say for most people, is that just including like healthy people?
Yeah.
Okay.
Like not immunocompromised people. People with immunocompromised status, fungal infections are a big problem, huge problem. So HIV patients that don't get their medications or have to miss it or something like that, fungal infections are one of the first things that shows up in your lungs, on your skin, that kind of thing. And they're really nasty, really bad, hard to treat infections.
Hey, Dr. Zong.
Yes.
So on the quiz, on the question that was like the subjective one, like what would we do? So for mine, I was thinking that because at the beginning of the paper and when you were talking about your research students, you were talking about how the database is so small and how I think you were saying like it's hard to like verify exactly what's going on or what you have sometimes just because the database is small. And so I was writing like that would be, in my opinion, what I would do. But you are the only mycologist I know, therefore the best mycologist I know. So
Woo!
what would you do?
I agree with you. I think getting our databases fixed is probably, or improved, is a #1 priority in my book. That's a lot of hard work. The other thing would be to change our methods a little bit, and instead of amplifying this small region, this ITS1, actually amplify a bigger swath of the DNA, right? So if you amplify all of the 18S and the ITS1 and the 5.8S and the ITS2, you've got more information, and you can actually build phylogenies. Now, this is crucial for fungal research because the ITS1 and ITS2 that we use as a barcode, they vary so much that it's nearly impossible to align them to make a phylogeny. And so, unlike bacteria where we amplify 16S and we can build a phylogeny from it, with fungi, we just have a little barcode that says it is this species or I don't know. Okay. So with bacterial work, if you get an unknown species, you can still place it within a group because you can build a phylogeny and you can say, oh, it's fitting in here with all these other, Shoagenella or whatever. And so you can make pretty strong case that you've got yourself a Shoagenella also, because it trees out with them monophyletically. With fungi, that's not the case, since ITS is not an alignable section of DNA. So switching to longer DNA read technology is going to clear up a lot of this. The current problem with long read DNA technology is that we get orders of magnitude fewer reads. So it's this trade-off. If I do what they're kind of talking about here, like Illumina sequencing, I can get 20 million reads on a single DNA run of my little code. And so I'm picking up all those rare species and everything. If I switch to this technology like nanopore technology or PacBio DNA sequencing, I can get longer reads, but I only get 30,000 of those reads instead of 20 million. And so I'm losing all the rare species that are in there. And so there's a trade-off. So I think those are my two kind of priorities, is just improving our methods so that we can forward with more direction, and when we do find unknown species, be able to at least put them into a phylogenetic grouping and see where they would belong.
So then if you spend so much time like increasing these databases, would that then go back to the fact, or would it be back to what we talked about before, that it's really hard to get funding for these kind of things, or that was a different concept?
No, I think it's still hard to get funding for that, especially if you write a grant proposal now saying, hey, I want to improve the database. The reviewers you get may say, that's not important. You should be doing this long read technology instead, right? And they may not agree that the rare species are worth finding. So it's really kind of a hard thing to sell. Luckily, here at UVU, we've got internal funding, right? So a few $1,000s here and there for student researchers. And so that's actually something I am working on right now, is trying to combine some of these databases, because we have like special database for human pathogenic fungi, and we've got the Unite database, and we've got one just for Fusarium because that's important for agriculture. And so what I'm working on is actually pretty cheap. It just takes time and computing skills, but putting together all these databases and then going and reanalyzing like 60 or 70 different studies. So it takes a lot of computing power, but in terms of funding, not much.
Okay.
Dr. Zone, I had kind of just a random question. I was curious. As far as like the fungal microbiome, does that have any correlation to like the bacterial microbiome and the hygiene hypothesis? Like you have more, then you could be more healthy or better immune system. That makes sense.
It does make sense. And again, I'm sorry, we just don't know yet. Most of the research labs that are microbiome, like human microbiome focused, for better or worse, it may be a good thing, but they do focus on bacteria.
Okay.
And so that's where we're getting all of our insights. Luckily, though, they're developing models for this kind of work. They're developing interesting hypotheses, and so fungal microbiome research can actually kind of piggyback off of what's being done in the bacterial microbiome world and use those new technologies and tools and stuff to kind of address these questions.
Okay, cool.
There seems to be, in my experience, and this is anecdotal, but like most of the fungal microbiome people out there are focused on plants, not humans.
Okay.
So I think there's kind of just a gap in expertise or interest in human research.
I had a question that might backtrack a little bit, but my internet's cutting out, so I don't know if it's been answered. But
Okay.
just like the fungi that like causes eczema and stuff, are we always carrying that around with us, and it just happens to like affect other people or not? Or okay.
Yep.
And does it have anything to do with like genetics as well? Because like my mom has psoriasis and my sister does, but I don't. So that would just be like those genes that would like affect
Yes,
it.
interacting with your microbiome. I mean, this is really complicated stuff to work out. It's never quite as simple as, oh, you have this fungus, it's met this critical threshold of relative abundance, and so therefore you will have this skin condition. You also have to figure out like your own genetics, what other species are around there interacting with that fungus. Okay, so it's high dimensional data that we're dealing with. It's never very straightforward, and we're just kind of fumbling around in the darkness trying to figure out things a piece at a time. Not to say we won't get there, but no single lab or researcher is ever going to be able to answer this on their own. It's going to take hundreds of studies kind of put together before we start seeing those patterns and noticing things.
Okay.
Yeah. But yeah, you've got malassezia on your scalp right now, I promise.
Yay.
This is kind of a random question, but is there any type of like antifungal drug resistant problem like there is antibiotic resistant problem?
Yep. And this has just been recent. As we've been spraying our crops with antifungal stuff more and more, we've now noticed that there are multi-drug resistant fungi, and some of them are actually human pathogens. So this has turned up with a couple fungal species just in the past 5 years. Outbreaks that we can't control, we can't figure out how to kill them, and they're resistant to all of our antifungal drugs. It's terrifying. Yeah.
Do you know if there's like any way to fix it? Because I know there's like been studies with phage therapy to help bacterial resistance. So would that ever work with fungal infections? Like,
maybe. I think that's interesting. It's only been literally in the past 5 years that we realized that fungi have their own little endophytes, and they've got bacteria living inside of the hyphae, and that those bacteria have like a strong viral component that's kind of shaping their community. And so this concept of mycoviruses is really new. Let's see, Jesse Euling is probably the world's leading authority on this. She's a brand new professor up at Oregon State University in Corvallis, but her research is still just getting kicked off. She's had some really interesting papers describing mycoviruses, but we're not at a stage now where we're much beyond characterizing them. We haven't moved into trying to manipulate them at this point.
Okay, cool.
Chandler had a question in the chat. She said, where do antifungal drugs originate? Like penicillin originates from fungi and lots of antibiotics originates in soil microbes.
Plants. They come from plants. Our best antifungal drugs. In particular, like in tropical regions, bromeliads, so things like pineapples, right? They produce really, really clever, like, antifungal compounds. And so there's actually a neat interplay between plant antifungals and carnivorous plants, because they're basically doing the same thing. Insect exoskeletons are made of chitin, and so plants that develop these secondary compounds to break down chitin and prevent fungal infection actually co-opted those proteins and used them to digest insects. And so carnivorous plants are really wacky, out there, fun source for antifungal drugs. I'm sorry, I'm having trouble seeing the conversation. Or it's like, it's got a real lag on my end.
No worries, I got you.
Okay. Online class is so much fun, isn't it? All right, so this paper, this was a review paper instead of a original research report type of paper. And it sounds to me like you all enjoyed reading it more. Review papers do tend to be more readable because they're taking a bunch of other papers, they've read them thoroughly, done their background, kind of followed the citation trail back, and they're putting together kind of a current state of knowledge in a topic. And so review papers can be fun for that. We'll have a couple more, we'll see, but not every paper you ever read is going to be a review. So some of them will be back into reporting original research. What are some other things that people answered for that quiz question on where you would go? What open questions would you try to address?
Well, I answered that I felt like it was necessary to look at the fungi that are commensal with us, but can turn pathogenic, and just be able to, because they seem like they should be pretty preventable.
Like if we could figure out the trigger that turns them pathogenic, we could prevent that kind of thing?
Yeah.
Uh-huh.
So that's kind of what my answer was, that like looking at those bacteria, or not bacteria, fungi, and making it more a common knowledge among doctors, which that maybe stemmed more from our lecture on Tuesday than it did from the paper. But just like, if it was something that was better understood, maybe that would kind of push it a little bit more to be well known in the medical field.
Yeah, and I'm not trying to disparage doctors. There's good historical reasons why they haven't cared about fungi in diseases. I mean, some of those are kind of ethically dark historical reasons, like we don't actually care about people who are immunocompromised or have AIDS because those outbreaks are happening in regions that are far away from Western science. Other reasons are just that fungi are in lower abundance than bacteria. When the germ theory of disease, came out, it was bacteria, not fungi, that people cared about, and so that's kind of just historical legacy of that's being what's studied. But also, bacterial diseases are more common and more life-threatening. Okay, until lately, when we start getting these fungi that are adapting to warmer temperatures because of climate change, meaning they are infecting people in new regions and more aggressively than in the past. So this will change. People will have to pay more attention to fungal diseases. But as far as microbiome work goes, this is really complicated junk. It really is. It's hard to understand. You get back all your sequence data, and making sense of it can be really difficult. There's just a lot going on.
So do you
Who
think
else?
that
Yeah.
just because, now we're starting to kind of learn more about it and it's, it's now become kind of more of a focus, is that why we're, starting to care about it more, I guess? And are we seeing more like fungal infections, or is that just because we know a little more about it?
It's both. We actually are seeing more fungal infections. They're on the rise. But we also now have the tools to notice the fungal microbiome, where before it just kind of wasn't on our radar. So now that it is on our radar, and has been for 10 years or so, science doesn't move that quickly, especially for something as complicated as this. And so I think there's a lag effect happening where there are quite a few labs working really hard to understand small pieces of the fungal microbiome in humans. And as they get results and publish those papers, it'll be a snowball effect, and we'll start developing more comprehensive theories of what the fungal microbiome is doing, how it's interacting with bacteria, how that's shaping our health.
So you think it's kind of just going to turn into more of a bacterial type deal where we just all of a sudden started learning more and then it just kind of exploded?
Yeah. And if you look at like bacterial microbiome papers, if you just do a Google search through Google Scholar and see how many hits it returns, by year, in 1995, there were a few dozen papers where people were like swabbing poop and growing stuff and counting the number of different colony types they found. And, last year there was probably 20,000 peer-reviewed papers on human bacterial microbiome. And so that's the case every year. It's just exponential growth in this area. Not all of them are groundbreaking papers, earth-shattering information or anything like that, but they do build on each other, and we are developing over time a useful set of images about what the bacterial microbiome is doing. Same thing is bound to happen for fungi. It has to. Otherwise, we're all going to die sooner than we normally would. You are all going to die, by the way. Don't worry. Anyone else have ideas for what topics in microbiome research would you like to see answered?
So I saw that there was a lot of comments about how Candida or some subspecies of the Candida genus basically show up everywhere in the human body. It seems like in every region that they cover, there's some form of Candida. And so I was thinking it would be really interesting to, it might be harder to figure out all of the microbiome, I guess, or figure out which, how all the fungi interact with our body, but I think it would be really interesting to maybe look at one genus and see if maybe we can figure out the relationship that Candida has with the human body, positive, negative, whatever, and study that. So I thought that would be really interesting to see as it's one of the more important fungus.
Right.
Body.
Yeah, going along with that, I also thought, because it mentioned that bacteria could be, my thing's cutting in and out, so it might have been mentioned before, but bacteria could potentially regulate the Candida genus. So just looking at, because when you take the antibiotic, that's when it causes the issues. So I thought it would be, pretty cool or important to see the relationship between the bacteria and the Candida, and if it regulates, so we can further understand what causes illnesses after taking antibiotics.
It's almost like we can't just study the relationship between Candida and the human body. Like, that's not going to get us anything useful, because the candida is also interacting with bacteria that are kind of changing its abundance or making it produce other secondary compounds, they might even be a trigger for why it goes pathogenic, right? And so I think it might almost be impossible to study that in isolation, like human plus candida, without having the other stuff in there. I think we would get misleading answers about the relationship of candida and humans if we didn't throw in all the things it was interacting with also.
And I agree with that 100%.
Yeah, okay.
Thinking that, yeah, no, that is not, I was not implying just that. I was just thinking more looking at that.
Yeah, focus on one species.
Narrowing down on one, yeah, exactly.
As a model.
Exactly.
And that's been a good technique for science in the past, right? We get a species that we can grow really well, you know, like fruit flies or yeast or whatever, and we use it as a model to study stuff and generate hypotheses. Then we can go back in and test those and work on developing a well-tested theory. And so maybe selecting one or two fungal species to focus on and try to figure out every aspect of them, you know, what bacteria they're interacting with, whether viruses play a role, that kind of thing. If we can get that figured out for one or two, maybe that will generate hypotheses we can go and test in other groups. Cool. Well, we're pretty much out of time. So any last words?
I guess I have a question. So we had to prepare the PowerPoint presentation, and I don't know if everyone has or not, but it would be really interesting to have access to those. Are you gonna provide those somewhere?
Yeah, I'll make them public.
Okay, because that'd be really cool.
Unless anyone objects, and then I won't make yours public. But I was planning on just sticking them on the website, like in week 6, and just say like student slides. Yeah.
Great.
All right. Well, sorry for any internet connectivity issues. We got at least 3/4 of the meeting recorded. And I will see you next week.
Thank you. Have a good weekend.